over 150 people online now between the panelists and the attendees, and the number keeps going up. So welcome back everyone to the public session of the EMSL Summer School. And we have a couple more great presentations this afternoon before we finish up for the day. I think especially for those of us who have been in all of the sessions today, our heads are spinning a little bit with lots of information. But it's been super exciting, and it's been fun seeing some of the pictures coming in that Andrea's been posting on our EMSL Twitter site. In fact, I wanted to share one that I thought was fun. We had one from Rebecca Prescott, who told us that she was traveling from Scotland to her home in Hawaii when she got stuck in Florida on a horse farm. So she sent us a picture of herself hanging out with a horse and participating in the summer school. So check out the Twitterverse and send us your pictures to Andrea if you want to share with us. So with that, I'm going to turn it over to Kelly and have her introduce the afternoon speakers.
Great, and I'll keep this short so we have max time, but I'm super excited about you guys being able to hear from both of these speakers. Our first speaker is Rekha Seshadri, and she is representing the JGI. Rekha does a lot of the stuff that we are actually doing this afternoon in terms of functional annotation and other things, and has a pretty impressive Google Scholar page herself. But what we've asked her to do is come here and speak on behalf of the JGI. And so we alluded to the role that JGI is playing in this Wonders program in terms of these community sequencing projects. But we also know a lot of you are early career, and so we thought you could really benefit from learning more about the DOE JGI capabilities directly from an expert. And I can tell you myself, and it's others on this panel who are also earlier in career can also tell you that they've really benefited from both JGI user support as well as EMSL support. So we really wanted to kind of end the day letting you know what kind of opportunities there are and for someone who doesn't have to be a metagenome person. The other speaker is Dr. Owen Brody from Lawrence Berkeley National Lab. And Owen Brody is going to really teach us how we can take like his tools and how we can take kind of the annotations and what these genes are or aren't and how to scale those into a much more quantitative framework. And I myself am super excited to hear this talk. So both kind of using omics in many of the tools that we use today, and you'll probably hear a lot of the same vocabulary, but giving you different kinds of information. And so with that, I'd like to welcome our first speaker. Rekha, it looks like your slides are up and you're ready to go. So do you want to just take over?
Great. Thanks, Kelly. Okay, the Joint Genome Institute, or JGI. Everybody hear me okay?
Yes, you sound great.
Okay, thanks. The Joint Genome Institute is similar to EMSL. It's a Department of Energy user facility. And basically, user facilities are funded by the Department of Energy Office of Science to enable access to large-scale science and advanced technologies and provide access to a worldwide community of users. So the JGI itself was established during the time of the Human Genome Project. They were actually responsible for 3 of the chromosomes as part of the human genome effort, and they were established in 1997 and currently located on the Lawrence Berkeley National Lab in Berkeley, California, and we have 280 employees. So our mission is to serve as a genomics focused user facility supporting DOE mission areas, which is bioenergy, carbon cycling, and biogeochemistry. So I had some back and forth with Kelly, and I've sort of contracted and expanded this presentation. So this is where I settled. I'm going to provide some information on JGI's user capabilities, so our portfolio of products. And I have some within parentheses because I'm not going to go over all of them. I just cherry picked some that I thought would be, most synergistic with the topics that you guys are learning about in the summer school. But there are definitely other capabilities, and I have links provided so you can check those out at your leisure. And then the second part is just talking about the user programs. And these are basically program calls where you can send a white paper and propose to use those capabilities. So you get free access to all the capabilities, the sequence data, and some of the computational analysis if you're approved, if you're an approved user. And then the third part of the talk is a user interface platform that we have for comparative genomics and comparative metagenomics called Integrated Microbial Genome. So I'll touch upon that briefly. And if I have time, if I'm not going over, I might do this primer on annotation since we've talked a lot about, KEGG and DRAM and, all the different pipelines that are used to do functional annotation. So I'm going to attempt to get there, but if not, these slides will be made available to everybody. So you'll be able to sort of hopefully learn just from looking at the slides later on. Okay, so capabilities, some of them. So as I said, we're here to serve sort of these DOE mandated mission areas, and serving those areas, we're divided into 5 different programs. We have the microbial and the metagenomic program. We also have a plant program, a fungal program, and probably the most recent addition in the last 5+ years is our DNA synthesis program. And really what I want you to take home at the end of the day is that we are definitely known for our DNA sequencing and our advanced DNA technologies, but we do have a lot of capabilities beyond sequencing. And part of the mission, the way I like to think about it, is that part of the directive really is to constantly evaluate and benchmark emerging technologies, which in our case has been sort of sequencing platforms, the software that goes along with it for the computational analysis, looking at other types of emerging technologies, and then passing on the access to those technologies and to the lessons learned and the know-how to our worldwide community of users. So sequencing is our bread and butter, and I would say that we have 2 primary platforms. This is not to say that we haven't evaluated or do not have other sequencing platforms, but these are our 2 primary platforms, which is the Illumina and the PacBio. A significant amount of data is generated from primarily non-human or environmental samples that arise from as I said, non-human environments. And each of these platforms is really applied to the products that are listed below each one. So I think there was a question earlier on today about long read technology and something about coverage. And I just kind of briefly said that, so PacBio in particular, we use for generating de novo genomes of both isolates, of both prokaryotic as well as fungal and plant genomes and such. And sometimes it's used in combination with Illumina-based sequencing. But we do have, we use the Illumina primarily for resequencing, generation of metagenomes, metatranscriptomes, and our single-cell sequencing. And the PacBio, on the other hand, is used for some of the products that are shown here. But as I said, we have many products that go beyond sort of the sequencing or these advanced sequencing products, like the single cell. We also have Methyl-seq, DAP-seq. I don't want to go on about different acronyms that you may or may not heard of, but we have a lot of capabilities. And please visit our product offerings page to see what all of those products are. But what I want to leave you with is that we have also capabilities beyond sequencing, which is our DNA synthesis program, for example, which allows the user to synthesize genes or pathways. If you're interested in functional characterization of genes and pathways, is very popular with biosynthetic gene clusters for secondary metabolites, for example. We also can synthesize combinatorial pathway libraries for metabolic engineering purposes. And we have individual program calls that allow you to use some of these capabilities. We also have metabolomics, and I thought that I would touch on that briefly because I think it might be of interest to this audience. Okay, so moving forward. I have some beeps happening. I hope my volume is still good. But so I just cherry picked one or two things to sort of focus on. And one of them is our brand new project, which is stable isotope probing metagenomics. And really the idea here is allowing you, the user, to link microbial identity to metabolic activity based on the uptake of isotope labeled substrates. So what the schematic is showing is that you would have samples from the environment that are probably incubated or enriched under certain conditions, and you provide then the isotope labeled substrates for its uptake, and the actively dividing members in that enrichment would then preferentially incorporate that label into its nucleic acid. And at this point, you would extract the DNA basically and send it to the JGI. And we would take care of the partitioning of the fractions and doing the quality control, and then it would run through our metagenomic sequencing pipeline. And as I said, this is a brand new project, and we're anticipating that we'll be able to accommodate 4 projects per year. Since it is brand new, I don't know if you can find any information through any of our program calls, but if you're interested in taking advantage of this opportunity and you want to learn more about the details or how to put together sort of your study design, please contact the 2 people that are responsible for bringing this particular pipeline to fruition, and that's Rex Malmstrom and Emily Elo Fedrosh, and that their emails are provided, and they'd be very happy to talk to folks if they're interested in taking advantage of this particular capability. As I mentioned, we have metabolomics, and this is all under Trent Northen's able hands, and through the metabolomics program, you gain access to LC-MS, MS-based metabolite profiling in both targeted and untargeted analyses. And again, I've provided a link. So please check this out at your leisure. I'll talk about the user programs. In particular, we have a user program that allows you to use JGI's capabilities complementarily with EMSL's user capabilities, and it's called the Ficus call, and we'll talk about that in a second. What it allows you to do is sort of submit one application to both user facilities and take advantage of the individual capabilities in both location under one study. Okay, so you can gain access, as I said, to all these products, some of the ones that I talked about, and an much more expanded set that I didn't talk about, by submitting a letter of intent. Generally, that letter of intent is really to check to see that the proposal that you submitted is of DOE mission relevance. I mean, it's really a check to see that it falls. It's not a study about human genomes or something like that, but it's of DOE mission relevance. And at that point, you're invited to submit a white paper proposal. And then those white paper proposals then undergo a peer review process by the scientific user community. And so, again, I'm listing a subset of these particular user programs. One of them is called the Largescale Community Science Program. And really the emphasis here is on large-scale projects. We have a cap of 3 terabases for Illumina sequencing and 100 GB for PacBio sequencing under the auspices of this particular program per project. And all the capabilities are offered. So the things that I mentioned, the DNA synthesis, the metabolomics, some of the advanced epigenomics, all of those capabilities are offered through the large-scale community science program. And as I said, you have those really large caps on data that you can reach for. And these are some of the specific theme areas that we've been encouraging proposals to sort of focus on. So I'm not going to read those out. I'll just point you to this, and this sort of gives you an idea of sort of the success rate of individual. And so the success rate for the large scale community science program is 35%. So in terms of the proposals that are ultimately approved, it's pretty good at 35%. The other program call that I wanted to highlight was the new investigator call. And here the emphasis is really to favor first-time investigators that have never had a funded proposal to the JGI before. So it really targets sort of early career scientists. And I guess the objective at the end of the day is sort of preliminary data generation. So it allows you to sort of develop a pilot project, and then you can use that information to then submit for a large-scale CSP. However, unlike the large-scale CSP, not all of the products are available under the new investigator call. It's limited to the sort of subset that I've shown before. So it still allows you to do the metagenomics, the metatranscriptomics, both for isolates, for microbiomes, and also the DNA synthesis and metabolomics product line is offered through the new investigator call. And then, as I already mentioned, we have a joint call called the JGI MSIL FICUS call. And please check out our website and probably MSIL's website for sort of timelines of when proposals are being accepted and when due dates are. But this particular call gives you access to capabilities at both facilities with just one application. And so I've listed, for example, for MSIL, we have high throughput proteomics, advanced imaging, and complex sample preparation. And at the JGI, it would be sequencing, DNA synthesis, and metabolomics with LC-MS based methodology. Okay, so one more about JGI. I've given you a link to sort of our user programs. You can explore all the different types of program calls that are out there. And I also wanted to mention that any investigator is eligible regardless of their institution or national origin. So we do have a worldwide community of users. All that we need is for it to be of DOE mission relevance. And if you have any questions about any of these user programs, please contact Tanya Woike. I have her email provided here below. She's serving as our interim deputy director for user programs, and she'll be very happy to chat with you about any questions that you might have about submitting a proposal to one of our user programs. Okay, so under the auspices of all these user programs, JGI has generated a lot of data. And what you're seeing here is JGI's contributions to public databases. And you'll see that a significant piece of the pie, both in terms of plant genomes and other types of microbial genomes, are contributed by the JGI. All of these data can be accessed to dedicated portals. So, for example, for plant genomes, we have something called Phytosome, and we have Microcosm for fungal genomes. And it's not just a repository for the sequence data, but it's also got tools for comparative analysis. On the prokaryotic side, for isolate genomes and metagenomes, we have something called IMG, Integrated Microbial Genomes and Microbiomes. And I'm just going to show you some of the some of the, not the tools. I mean, it's a large behemoth, so it's kind of hard to cover it all. But I just want to make you aware that this tool exists, and it's a data management system for comparative analysis of sequence data. So basically, all of this JGI generated data does end up in IMG. So we have a dedicated assembly team, because we are a sort of high throughput, high production type environment. We have hundreds and thousands of samples that need to be assembled and annotated. And all of those data get assembled and then annotated through the IMG annotation pipeline. So you'll see here, data sets that were generated by the JGI. For isolate genomes, we do go to GenBank and periodically update public isolate genomes from GenBank or from RefSeq. In terms of metagenomes, we have, again, primarily JGI generated data, but we also have public metagenomes, things from the Human Microbiome Project, for example, and some other genomes. It's not everything that's in GenBank, but there's still about 23,000 metagenomic samples. And we also have these other uncultivated entities, if you will, like the single particle sorts, cell enrichments, and metatranscriptomes. Okay, so there's a lot of data that you can look for, explore, and there are tools for comparative genomics. We conducted a webinar series relatively recently, and so all of those videos are available if you're interested in checking out what the possibilities are. Again, something that I wanted to highlight, because we're talking about metagenome bins this morning, is for all publicly available samples, all publicly available metagenomic samples, the initiative is to provide sort of pre-computed metagenomic bins. And this is the schematic that shows you that we run MetaBat to generate these bins. We do a certain amount of QC to remove sort of offending contigs that don't fit in that pre-computed metagenome bin. And we perform a CheckM based evaluation, just like you did. And only the high quality and the medium quality metagenome bins are then loaded into IMG. And so at present, we have over 101,000 bins that have been predicted from over 12,000 metagenomic samples. And all of these bins can be retrieved or explored based on their taxonomic affiliation, based on both an IMG's centric methodology, as well as your genome taxonomy database that you used probably this morning, GTDB-TK taxonomy, or by their underlying environmental or ecosystem metadata. And then you can do sort of these cross metadata searches using a bin search tool. So here's just a screenshot of what it looks like. So I'm showing a screenshot where I've sort of navigated down to 152 metagenome scaffold bins assigned to the genus Nitrosopumilus under the Thaumarchiota. And you can click on this number and it'll retrieve a list of all those bins, along with a lot of information about the content of those bins and the CheckM statistics. You can also peruse the bins by their ecosystem classification. So in this particular example, I've drilled down from terrestrial samples down to permafrost, and it's telling me that there are 768 bins that have been called from samples that are attributed to permafrost. And as I mentioned, we have a webinar series. I have links to all of these in the PDF document that I think you will be, either you already have or will receive shortly. So check out the webinars, and maybe it'll be of some value. Okay, I don't know how I'm doing on time, guys. I'm going to assume that I'm still good, and I'm going to soldier forward unless somebody tells me to stop.
Just about 9, well, yeah, it's probably 6 or 7 minutes if you want to leave a couple for questions.
Okay. All right. So other than JGI sequence data, we also encourage people to submit their own metagenomic data. So whether it's sequenced by the JGI or not, we do accept submissions within IMG of external data. So you as an external submitter might have assembled something in KBase, for example. You can submit those assemblies to the IMG annotation pipeline. Here's some details about what you would need in order to do that. And here's just a schematic of the IMG annotation pipeline. I don't want to delve into these details. What it gives you on each side is this is the structural annotation pipeline. So it's the gene calling. I think in the morning, people mentioned Prodigal. This is the structural annotation pipeline where the open reading frames are called. We use 2 different gene calling methodologies. There's a standard operating protocol also that's available. And then this is the annotation pipeline. So it gives you an idea of the tools and the databases that are used to do the structural and the functional annotation. And then all of that content then gets submitted to the UI system, and it's then sort of presented there in sort of a comparative context and in the context of various tools where you can do some comparative analysis. So here's, I guess, the primer part. I just wanted to briefly talk about structural and functional annotation. Just to familiarize everybody, although you've learned this before, so I'm just going to go over this quickly. So the purpose of structural annotation is to get at protein coding, at coding sequences, not just protein coding. It can be regulatory or RNA sequences, tRNAs. So structural annotation really is just looking for the features of a gene. So looking for canonical starts and stops, and looking for signatures or ribosomal binding sites, and calling these open reading frames in all 6 directions, right? In all 6 frames. And the ultimate purpose of structural annotation is to find the coding sequence. So structural annotation is not open reading frame finding, but the purpose is to find coding sequences. And in prokaryotic genomes, this is relatively straightforward. We try to go with the longest open reading frame that's usually correct. And you try to resolve any overlaps. In certain high GC genomes, this can be an issue, but that's the purpose of structural annotation. But when people think about annotation, they like to think about this part, which is functional annotation, where you're getting from sequence to an idea of the enzymatic function, an idea of the underlying biology. And so to do this, you search various types of databases and you look for similarity. And based on that similarity, you transitively assign a function. And so there are many, many archives of databases, and you've heard about a lot of them today. So I'm going to skip this part. It's just talking about how you have bioinformatics curators around the world sitting around and trying to record information from publications in a structured way that can be represented in a database and that we can then use to do our comparisons. Searching these databases, there are 2 general methods. One is everybody's favorite BLAST, which is called the Pairwise method. And all that is you have 2 strings. In this case, it's amino acid string sequences, and you map these 2 strings, allowing for certain substitutions, a certain amount of insertions and deletions, and then you use kind of this best hit approach to parse the information, and then you transitively assign function based on that, and BLAST is an example of that. But there are many other tools that are being developed. As the size of databases grow, there's always a trade-off between sensitivity and speed. And so some of these other tools that you may or may not have heard of try to solve that problem. And databases that you do pairwise searches against are obviously NCBI NR. I'm sure everybody's been there at one point to run a BLAST job against their NR database. So within IMG, we also have an IMGNR database that we do a pairwise search against. And as Dram already mentioned, we also do a search against the KEGG orthology database. So here's a pathway map for that. The second method for looking for similarity is this HMM search that you heard about this morning. These are profile-based methods. And the difference between these is that first, the curator develops a really good alignment. So you take a bunch of reference sequences that are well characterized, well known, generate a really good alignment, and then compute the statistical probability for the amino acid in those positions in the seed consensus. And so here's a schematic again. You have a multiple sequence alignment, and then you have the consensus, and you compute the statistical probability, and you build a profile such as a hidden Markov model or a position-specific scoring model. And hopefully everyone can appreciate now that this is computationally a lot cheaper than having to do a blast search against an entire IMGNR database or an NCBNR database. Instead, all you're searching is these consensus models. And it also can be more sensitive than a pairwise alignment. So very frequently, you might not get a pairwise hit against an NR database, but you might get an HMM search result. And so again, there are many different databases that are established and updated that are out there. And here are some examples in addition to the ones you heard about in the context of RAM. So within IMG, we perform HMM searches against NCBI's COGS database, the TigerFam database, the PFAM database, and then more recently, we've added Cat FunFam, Smart, and Superfamily databases. Okay. You might ask, why do we have so many different databases? And maybe somebody's already answered this question before. They're not all perfectly overlapping. They have individual strengths and individual limitations, if you will. And so there is a reason why we try to maintain annotations, all of these different types of databases. And the reason for that is sort of maximize the opportunity to make an inference.
It's 4:23.
Okay, thank you. I'll wrap up. So all the data can get submitted to the IMG annotation pipeline, and on the UI side, you have access to various tools that allow you to explore your data and do some comparative analysis. I will leave with this slide. Just to let you know, we have an annual user meeting. Obviously, we didn't have one this year, but I think we should be back. Well, we were all fingers crossed we will be back on track next year. And that's an opportunity to hear about exciting new projects from our users and from the scientific community at large that use sequencing-based approaches. IMG has its own hands-on on-site workshop that'll be here at Lawrence Berkeley National Lab. And we have a webinar series that you're welcome to check out. There's a YouTube channel that has, I think, eight videos posted there. Okay, I'm going to stop. I apologize if I went at a breakneck speed. I'm assuming there'll be questions. I don't know if we have time to take them now, or I can try to answer them via chat.
So yeah, we can definitely, you can look over the chat, but one of the key questions we can answer real quickly was, well, a couple were related to access for users. And so one you already answered very well in your presentation. One user asked before your slide about international users and if it was open to non-US scientists. And so clearly the answer to that is yes. Another one asked about supporting projects funded by DOE agencies other than BER. So for example, ERE, the renewable energy or fossil energy offices. And if you could maybe give a little insight on that.
I think the answer is yes. Perhaps I'm not the best person to answer that question. Tim, I don't know if you know more than I do.
Well, I think, and Nancy answered in the chat too. I think it's really an issue of, like you said, for the international or anybody else, as long as it's aligned with BER and DOE science missions, that's really the issue, right? So, obviously, if it's not aligned with those missions, the answer is no. But if it is, it really doesn't matter so much who's funding the actual project as much as it is about the alignment. And I think that's also true for EMSL as well. So, and obviously, thanks for pointing out the joint EMSL JGI call. That's, I think, been a really successful avenue for users to be able to access both facilities with one proposal. So